In this video, I will be showing you how to do the streak plate method. Before we begin, gather up all the tools that you will need for this experiment. For this experiment, you will need gloves, paper towels, germicide to clean up your workspace before and after your experiment, goggles to protect your eyes, plastic petri dish which will stand in for your agar, a lab coat, China marker, test tube, inoculation loop, your stock culture, and your Bacto incinerator. Before you start your experiment, make sure to clean up your workspace using germicide and then wipe it down using paper towels. Once you've gathered all your tools, pick up the plastic petri dish and label the bottom of your plate using your China marker. You want to put your initials, and you want to put the letters SM/SA. These stand for the bacteria that is within your mixed culture. Once you're done labeling the plate, create the following pattern with your China marker on the bottom of the plate. Then flip the plate back over so that the top lid is facing up. You can kind of see that you're looking through the agar and you can see the pattern. It's like in reverse. This is what it would look like. Then obtain a mixed culture, also known as your stock culture. Before you can actually get any of your stock culture, you want to sterilize your inoculating loop in the Bacto incinerator. Then you want to allow your loop to cool. We have to allow this to cool because of the bacterial cells. Some cells may be heat sensitive, which means if the loop hasn't cooled down, it could kill these bacterial cells and we won't get a proper result. Now that your loop has been sterilized, you can obtain some of the culture by scraping it out using your inoculating loop. Since I don't have a test tube rack, I'm just going to be placing it down on my bench top. Now you are ready to do your streaking. Open your petri dish in a clamshell-like fashion. Streak the loop 3 times across the surface of the plate. Just follow along with the drawn-in lines. Remember, you can only go from left to right when streaking. Then you want to re-incinerate your loop. We do this between each passing/spreading of cells because A, it ensures that your loop is clean, and 2, it reduces on the number of bacterial cells that will be spread or streaked into the next quadrant. Now go back to your agar plate and then spread some of the culture that was added to the plate downward across the plate in 3 streaks. Again, just follow the drawn-in lines and only go in one direction. Incinerate your loop again. Again, go back to the agar plate and spread some of the culture downward, again along these lines. Incinerate your loop again. Just continue to repeat this process until you've completed the pattern drawn on the plate. Incinerate your loop. And for the last time, go back to the agar plate and spread some of the culture downward again along these lines. Then incinerate your loop one last time so that it is sterilized before you set it back down on your table or workbench. Once you've completed all of these steps, Incubate your specimen at 37 degrees Celsius for 48 hours before observing for colonial growth. After that, you are now ready to decontaminate yourself and your workspace. First, dispose of your gloves in the biohazardous waste container. Your gloves are now contaminated from the experiment that you have just conducted. After you've done that, wash your hands thoroughly. Then use germicide to wash down your workspace, and then use paper towels just to wipe it up and dry it off.